Hey everyone, my name's Christina and this video is on hexokinase and glucokinase. So imagine you're on your family medicine rotation. You finish rounding and charting for the day and now you're sitting in the residency didactics. Today they're going over diabetes and specifically insulin-dependent diabetes. Midway through the lecture, one resident notices how sleepy the medical students look and decides to ask, what enzyme does insulin act on? All the other medical students look dazed, trying to recall M2 Pharm, but not you. You recall clearly, and you raise your hand to be called on. In this video, we're going to be talking all about hexokinase and glucokinase. In the next few slides, you should be able to 1. Recognize the importance of a hexokinase and glucokinase in the metabolism of glucose. 2. Describe the location, affinity, and speed of each enzyme. And 3. Associate relevant clinical disease with hexokinase or glucokinase mutations. Alright, let's dive right into the video. Do you remember what the function of a kinase enzyme is? A kinase enzyme transfers a high-energy phosphate group from ATP to a substrate. So we have a cell here and a molecule of glucose located outside the cell. It is free to enter and leave the cell in its current form. But once a glucose enters a cell, it meets with the enzymes hexokinase or glucokinase, which adds a phosphate group to the glucose molecule. Thus, the first step of glycolysis is phosphorylation of glucose to create glucose 6-phosphate. This step requires 1 ATP. Why is a phosphate added to the glucose? Well, in addition to preparing glucose for the next step in glycolysis, this also gives glucose a negative charge, which now prevents it from passing through the plasma membrane, which effectively traps the glucose within the cell. As stated earlier, this reaction can be catalyzed by either hexokinase or glucokinase. The difference between these two enzymes have to do with their locations and enzyme kinetics. You will learn more about enzyme kinetics in the pharmacology section, especially about the Km or affinity and the Vmax or speed, which you see here in this graph. For now, I'll briefly go over the kinetics of these two specific enzymes, hexokinase and glucokinase. First, we'll focus on hexokinase. It's found in almost every cell type in the body, except for liver cells and pancreatic beta cells. Hexokinase has a high affinity for glucose, but has a low maximum reaction rate. This means that the concentration of glucose needed for this enzyme can be used is relatively low since it has a high affinity, but the speed at which a reaction occurs is pretty low due to its low Vmax. Hexokinase is not affected by insulin, but does receive negative feedback from high amounts of glucose 6-phosphate. So basically, when the cell has low glucose 6-phosphate levels, hexokinase will catalyze any glucose available in order to form glucose 6-phosphate and store it within the tissue. However, if there's a large amount of glucose in the blood, such as after a meal, hexokinase won't be able to keep up since it has a low capacity or low Vmax. Okay, so on to glucokinase. Glucokinase comes into play after meals when there are high glucose loads. Where is glucokinase found in the body? Glucokinase is only found in the liver and in beta cells of the pancreas. This enzyme has a low affinity for glucose but a high capacity or reaction rate. This means that glucokinase is not going to be active unless you have a lot of glucose, but when you do have a lot of glucose, it can act very quickly to phosphorylate it. Also, it has no feedback inhibition from glucose 6-phosphate, so as long as there's lots of glucose in the bloodstream, glucokinase will keep phosphorylating glucose inside of cells. It does, however, receive feedback inhibition from fructose 6-phosphate. Glucokinase is also directly influenced by insulin. You can think of this as being kind of a buffer system because after you eat a large meal, glucose will activate the beta cells in the pancreas, which will then release insulin, further activating glucokinase in the liver so that the glucose is removed from the blood and stored as glycogen. Another way to think of these two enzymes is like the tortoise and the hare. Hexokinase is the tortoise because it acts at a slow and steady pace, while glucokinase is the hare, speeding up and slowing down depending on the amount of glucose in the blood. I want to touch on the concept of diabetes, and specifically maturity onset diabetes of the young, or MODY. Again, you'll learn more about the disease diabetes as a whole in the endocrine section, but in a nutshell, diabetes results in an increase in glucose in the bloodstream, and there are many different forms of diabetes. type 1 versus type 2, and then MODY. Maturity onset diabetes of the young is a group of monogenic diseases, meaning there's usually only one mutation in one of the few genes causing the diabetes, versus the other forms of diabetes which usually have multiple genetic or non-genetic causes. One of the mutations in MODY can involve the glucokinase gene. This mutation will functionally alter the glucokinase enzyme, producing a form of diabetes. Now, why is that? Remember, glucokinase is induced by insulin when there are high levels of glucose in the bloodstream. If a point mutation causes a glucokinase enzyme to malfunction, insulin will no longer activate glucokinase like it normally would, resulting in increased levels of glucose in the bloodstream because glucose would no longer be trapped inside the cell. It's important to note that hexokinase has not been associated with MODY. Can you think of any diseases that involve hexokinase? One such disease is essential fructosuria. Now, we just went over how hexokinase and glucokinase play a role in glycolysis through phosphorylation of glucose 6-phosphate, but this specific disease involves the metabolism of fructose. It is due to a defect in the enzyme fructokinase, not hexokinase itself. Fructose is typically first converted to fructose 1-phosphate via fructokinase, and will eventually enter glycolysis. But in this disease, fructokinase is defective. So if fructokinase is not functioning, then fructose is metabolized by hexokinase, which converts fructose to fructose 6-phosphate, which can then proceed through glycolysis. This is a rather benign disease, and clinically, you really only see fructose in the blood and urine. Don't forget to check out our video on fructose metabolism. Okay, let's do a quick flash quiz. Piggybacking from our introduction scenario, a 65-year-old with type 2 diabetes injects insulin before his lunch. How does insulin affect the action of hexokinase and glucokinase? Hopefully you remember that insulin induces the enzyme glucokinase, but doesn't affect hexokinase. This is because hexokinase is always active, while glucokinase must be stimulated by insulin when glucose levels are high in order to create more glucose 6-phosphate to trap glucose inside the cell. Okay, so the bottom line is, hexokinase and glucokinase are involved in glucose metabolism by phosphorylation of glucose to create glucose 6-phosphate. Hexokinase functions in almost all tissue, except the liver and pancreas, while glucokinase solely functions in only the liver and pancreas. MODY, or mature onset diabetes of the young, is a subset of diabetes due to a defect in glucokinase. Essential fructosuria is a disorder of fructose metabolism that results in hexokinase functioning as a main catalyst in metabolizing fructose through glycolysis. Alright, that wraps up hexokinase and glucokinase. Thanks for watching. If you enjoyed the video, don't forget to give a thumbs up.